Good evening, viewers. Welcome to the new session of the Pre-Med Pro, and today we will continue with the mRNA as I have said yesterday. And in 2 to 3 lectures, I'll complete this whole chapter and I will provide the mind maps of this chapter. Okay, so it's a very short video, about 10 minutes, approx 10 to 11 minutes it will take, and I will complete the mRNA and tRNA in this lecture. Give the heading as mRNA. mRNA, let me draw a picture first of mRNA. Now see, here one end is this end is the 5' end considered, and this frame is the 3' end. Okay, now what happens, these are the codons. Okay, these are the codons, and now okay, this one and this one, the first two codons are the initiating codon, initiation codon. Okay, and so the last two one will be a termination codon, Okay, now what happens, for the mRNA we require one ATP. One ATP is required. So what's the function of the ATP in mRNA? It activates the amino acid. Amino acid. One GTP is required. For the sliding. Its action is for sliding. And one GTP is required for tRNA bind at A site. Binding of tRNA at A site. A site means acylation site. Okay, so now this is the initiation codon, and okay, now this one is UTR. A and A and A. Okay, now what happens? What is UTR? Question may arise, what is the UTR? UTR. So UTR is untranslated region. Untranslated region in mRNA. Untranslated region in mRNA before the initiation codon and often after termination codon, and often after termination codon. Okay, so it's a region before the initiation codon and after the termination codon. Okay, now what happens, protein are not formed here. Protein is not formed here. Okay, but they makes translation efficient. Efficient translation is seen here. Okay, now what happens, in prokaryotes, what happens in prokaryotes? Initiation first codon. Okay, now it happens as this is the 3' and this is the 5'. Okay, the RNA is just one folded, and now the codon is here AGG, and let it be the codon here is again AGG. Okay, and here the codon is UCC, and here the codon is again UCC. Okay, now what happens? It is a 30S RNA because it is an of eukaryote. Okay, it is 30S to 60, that is it contains 16 S RNA. Okay, now the function of the sRNA is clear, function of the mRNA is clear as of now. So here we can write here the shine del grano sequence, or you can say SD sequence is seen. If the question is made tough for separating the students for the AIMS JIPMER, so these questions can be asked. Elsewhere, these are the most toughest question, and I think the 99% of the students will leave these MCQs. Okay, this year you will not be able to, very few candidates will be able to mark up with 720 marks, because NT will take the precaution so that a very less number of students can achieve it. Okay, now going to the next part, it's of tRNA. So please make note first of tRNA. It is a 2D secondary structure discovered by Robert Holly. Okay, he named it also a clover leaf structure, clover leaf model. लवंग होता है ना, लवंग, उसके जैसा दिखता है tRNA. इसका सेकंड है 3D structure discovered by Kim, and it is of inverted L shape. Okay, the third one is single stranded but folding. And the fourth one is, so it appears, as it is a single stranded but folding, so it appears as a double strand. Okay. Now the 5' end, it's of guanine. The 3' end ends with CCA sequence. Okay. Now some abnormalities can be seen in this region. Let me draw it. Okay, here a short arm. It is a 5', it is 3'. Amino acid receptor site, ACC So adenosine in sugar changes to third degree carbon, and the third degree carbon changes to OH group. Okay, now this OH group is attached with amino acid binding. Okay, here now on this prime 5' end, guanine is there, and this is called the DHU loop. This is the DHU loop. Okay, this is also called dihydrouridine, actually the dihydrouridine loop or the DHU loop. Some abnormal bases can be present here. Some abnormal base present, pairing not possible. If some abnormal base is present here, pairing is not possible. Loop, so possible loop need to be formed. Okay, now here, and this is an anticodon loop. So in the NEET PG, I have seen a question that which RNA may contain a thymine group. If the question comes in NEET UG, this is a question came in the NEET PG, which RNA contains the thymine group, and you can say it the tRNA, because the tRNA at the anticodon side there is a psi that defines a thymine group may be present on that RNA. So if a question comes because of RNA containing a thymine group, you can say it's a tRNA. Okay, this is the anticodon part. So this structure was given by, this is a 2D structure and given by Holly. And in this loop, in this is a T psi, that is pseudo-uridine, contains pseudo-uridine as well as ribothiamidine. This one contains the thymine group, ribothiamidine. So if the question comes which RNA may contain a thymidine group, you can say a tRNA. Okay, so now till then I have completed the mRNA and the tRNA. So the RNA world is completed here. In the next day I will start from a new topic, and this is a very short video as I have told yesterday, so that you can get it revised in a very short span of time before the examination. So till then, good night. Please share and subscribe this channel and write the comment. And so how I am going to teach and how I am teaching you, how it is being affected in your studies. So you can write it in the comment box, and please share and subscribe it. Thank you. Good night.